The effect of acetoin found in vapes/e-cigarettes on calcium mass by Charlotte Talbot. According to the Center for Disease Control and Prevention, smoking remains the number one most preventable cause of death in the USA, responsible for approximately 480,000 fatalities each year. While cigarette usage has declined, vaping has rapidly gained popularity, particularly among high school students. In fact, approximately 25% of 12th graders report vaping. One major health concern of vaping is the addictive chemical nicotine, which can cause numerous health issues, including respiratory and cardiovascular problems. Additionally, nicotine has been linked to osteoporosis, a condition that leads to weakened bones. In light of the growing use of e-cigarettes, one of the concerns is how the additives in vapes, such as flavoring chemicals, affect the body. Specifically, I wanted to investigate how acetoin, a flavoring chemical commonly found in e-cigarettes, affects calcium carbonate precipitation. I hypothesized that there would be a strong correlation between the amount of acetoin in the solution and the amount of calcium carbonate precipitate formed. In this experiment, I used a lime water solution, otherwise known as calcium hydroxide, as a model for calcium in the human body, and I added nicotine and acetone to mimic an e-cigarette solution. Each solution had 40 milliliters of nicotine and various amounts of acetone. The acetone concentrations included were 0 milligrams, 529 milligrams, 239 milligrams, and 64 milligrams, all of which are common amounts found in an e-cigarette. Each of the concentrations were tested 3 times for a total of 12 trials. After each solution was prepared, carbon dioxide was bubbled into the solution, leading to the formation of calcium carbonate, which precipitates out of the solution. A vacuum filtration was used to separate the calcium carbonate from the rest of the solution. The amount of calcium carbonate was measured by weighing the filter paper before and after filtration. The control group was just nicotine and lime water without any acetone, while the experimental trials involved adding acetone into the solution. The independent variable was amounts of acetone, and the dependent variable was the mass of the calcium carbonate. After the data was collected, linear regression was used to analyze the trends between the variables. As seen in Figure 3, the mass of calcium carbonate precipitate decreased as the amount of acetone increased. Figure 4 shows a similar trend with a visual representation. The 0 milligram acetone condition resulted in the most calcium carbonate precipitation with an average of 0.39 grams, while the 529 milligram acetone condition showed the least precipitate at 0.19 grams average. Linear regression analysis showed a possible negative trend between acetone concentration and calcium carbonate mass, with an R-squared value of 0.708 indicating some correlation, but not statistical significance as there was overlap in the error bars, which suggested variability in the data and highlighting the lack of significant results. While a negative trend was observed, the data did not provide conclusive evidence to support the hypothesis. The lack of significance may be due to experimental limitations, such as variability in the lime water solution. A possible explanation for the trend found could be that acetone may act as a chelating agent, binding to calcium ions and potentially reducing the amount of calcium carbonate that forms. This is similar to how other substances, including nicotine, reduce calcium absorption in the body. A major limitation was the variability in the lime water solution because the calcium hydroxide needs to be precisely measured and any inconsistencies affected the final result. Future research could include repeating the experiment with more consistent preparation of the lime water solution, which would then yield more reliable data. Performing more trials would also be useful to obtain more accurate data. The conclusion is that although there was no significant evidence to support the hypothesis, the trends observed suggest that acetone could have an effect on calcium mass. Further research is needed to confirm these findings and explore the potential long-term health implications of acetone and other e-cigarette additives. References. Thank you.